talking today about the gene expression. What is the meaning of gene expression? It's how to transform the information of the DNA, okay, to give a protein. So it will be expressed into a protein. And the process actually is called protein synthesis. So gene expression, it has two steps. The first step, transcription and the translation. The first one, transcription, it happens in the nucleus, and this is the nucleus because actually the DNA inside the nucleus. So the DNA will give a complementary copy of a part of it, only a small part, it's a gene, and it will give only one strand of that part will give a complementary mRNA. Then the second one, translation, we will translate. Here it's outside the nucleus, still in the cell, but outside the nucleus. This mRNA will be decoded into protein. So the codes carried here, the mRNA, will be translated into protein. And this code came from the DNA. Originally came from the DNA. So this part, the code of this part, will be giving this copy. Then mRNA. The mRNA can leave the nucleus. So it will leave the nucleus to be translated into a protein. So the code of the mRNA will be transformed or translated, decoded into protein. Okay, to sum it up, the first step is transcription, and it's all about um, getting an mRNA copy from the DNA molecule. Not all the DNA molecule, only the part, maybe this part, let's assume that this is the part that I need, the gene, and only one strand of the DNA in that part will be or just giving that copy of the mRNA, complementary copy. Why it has to give a complementary copy of the mRNA? It has the DNA information. Why the genetic information? Why it can't go outside the nucleus and just give this information to the ribosome to convert it into or translate it into proteins? Because the DNA is here. The DNA is in the nucleus. This is the nucleus. The DNA is in the nucleus and it can't leave the nucleus. That's why it uses. So this is the copy of the RNA here, and the RNA will just go outside the nucleus, I mean, to the cytoplasm outside, to the ribosome, and here the second step, the translation, will be done. So again, what do you know now about the second step, which is translation, the second step? Only what I know now is the mRNA will leave the nucleus and it will go to be decoded into protein. So the genetic code of the mRNA which came originally from the DNA will be coded into protein. So this is what I know now about the translation. Now we'll start the steps. The first step and second step, transcription and translation, but in details. Now I'm going to start the stages of transcription. Transcription, we said the DNA, this is D, okay? The DNA will be transcribed into RNA, actually mRNA, messenger RNA. Why it's M messenger? Because actually I'll be sending it because I am the DNA. I can't leave the nucleus. So I'll send a messenger to the ribosome with my information. Okay, so the DNA will be the template and the RNA will be the copy from that template. So the helicase, it's an enzyme. Just now take a paper and write down number one, the first enzyme that is involved in this process, the transcription, is helicase. The helicase will unwind the double helix. So easily I can transcribe the part that I want to. So number one, the helicase will unwind. Then the DNA, the RNA polymerase will attach to that part I want to start to transcribe it. And I'll tell you what is the RNA polymerase. You know it from the DNA transcription, right? The previous lesson. And it's actually responsible for adding the nucleotides together to form the copy that we need. Here we need the RNA. But how can this helicase and RNA polymerase, how can they know that this is the gene that I want to start transcribing it? From a promoter. The promoter is a sequence of DNA, like some nucleotides, and it shows that the RNA polymerase has to come here and attach to that part to start transcribing that gene. Each gene has its own promoter. Okay, so each gene has its own promoter. Then we have the second stage of transcription, which is elongation. So the first one was initiation, that I started the whole process by unwinding the two strands. Here the DNA, the double helix became unwinded. Okay, so it's easy to form the RNA. Number two, I added the RNA polymerase to the DNA. How did I know that this is the part that I have to start from? By the promoter. It's like a sequence in the DNA. Now the elongation, that I started already adding nucleotides, as you see here, and the red one is the RNA, the uh, the copy of the RNA, and the elongation is increasing the length of that RNA copy. Then the strand will be increased till I have the whole part that I need. Don't forget that it's complementary. The RNA is complementary to the DNA that I need to translate later. So here I add complementary RNA nucleotides complementary to the DNA nucleotides that I have already in the template. So G, C, A, U. You understand? So again, just to make sure that you understand that this is the RNA strand and it's made from this strand of the DNA. It's the template that I refer to add the new one. I refer to an old one, the template, and it's complementary. So the T here, the corresponding one, it has to be A. C, the corresponding one has to be C. C, the corresponding one has to be G, and so on. Okay, and here the RNA polymerase, the one adding the new nucleotides of RNA. Then the termination, the last one is termination. So I have initiation, elongation, and termination. Those three, they are the stages of the transcription. Okay, then the RNA here reached the stop signal. So stop, I don't need any more transcription, and this is the all RNA strand that I need. So the RNA polymerase will deattach. The process of transcription is done, but the mRNA is not ready yet for the translation. Before it leaves the nucleus, the introns will be removed from the mRNA, and the exons will be connected together to give the final mRNA. If the introns are not removed, so the resulted protein will be non-functional. It will not be that protein that we want, because actually some parts will be giving um, unwanted results. Okay, so we have to remove those parts that we don't need, and we have to connect the part that we need, so it will be the one we want it to be translated, and this one the one ready for the translation now. I'll stop here just to remind you before starting the translation that this is the base pairing. Okay, this is the DNA, and this is the other one, the other DNA strand, and here G is with C, T with A, and this is for the DNA. But for the RNA, just let it with the other color, the A, so the RNA, the A with U, but the G still with C. So this is the base pairing. This is the complementary. So this is the template, and this is the complementary DNA. In our case today, it's the template DNA, but the complementary strand is the RNA. That's why I just reminded you with the base pairing of DNA and RNA. Now we're ready to start step 2, translation. Step 2, translation, it happens here in the ribosome. This is the ribosome. The ribosome, if you remember, it has a big unit, a large unit here and a small unit down, and it has, inside it has two sites, 1 and 2, for the tRNA to start translation. For explaining the types of RNA in depth, so I want you to meet the types of RNA. You know some of them, but some of them are new to you. So I'll introduce number 1, mRNA, and we said it's messenger mRNA because it's actually a messenger. It takes the information, a copy of the information, a complementary copy of the information from DNA to be translated in the ribosomes, the protein site, into proteins. Amino acids, then proteins. Number 2 is the rRNA, the ribosomal RNA, which is the ribosome itself. The ribosome has two parts of two units. The big one is up, the large unit, and the small unit down. And actually between the two units, the protein will be synthesized. This is the site of protein synthesis. So the mRNA will just go in between, another color here just to be obvious. So here in between the two units, the mRNA, it will be read by the ribosome and translated into amino acids. Each 3 nucleotides, for example, GCA. So GCA will be here translated to an amino acid. Who will bring the amino acid? Mr. rRNA, the transfer RNA. It transfers the amino acids to the protein synthesis area. Okay, so the 3 mRNA, which is named the 3 nucleotides in the mRNA, GCA, for example, it's a codon. Each 3 will be a codon, and it will be translated into an amino acid, and it will be brought by the tRNA. to explain more about the tRNA, it's actually, here the most important one. The codon, it will be the three nucleotides of mRNA. How can I know that this GCA, for example, they match the broad amino acid that it was brought by tRNA by the anticodon? So we said the codon, here the codon GCA, GCA, the codon in the mRNA. It will be matching an anticodon, that purple one here. So it will be complementary. G is C, this is C, okay? And C is G, just to make sure that it's a G, and A with U. So this is the anticodon in the tRNA. This CGA is the one will bring the amino acid here. So all the tRNA that has the anticodon CGU, it has this particular amino acid. Okay? So that's how we match the codon or the sequence in the mRNA and the amino acid, the broad amino acid by the tRNA. And this is actually the key of the whole lesson. So if you understand this one, you can just relax and go to the next lesson, the code, how to get the genetic code easily. The first stage of the translation is initiation. Initiation, the first stage of everything, because it means the start of the process, okay? So the first step is initiation, and it's actually when the mRNA binds to the ribosome, the ribosomal unit, the small subunit. So the small one is the one down, and the large subunit is the one up. Okay, so when the mRNA binds to the small one, it's the initiation. Then the first codon, it's always the first codon, always the start codon. The start codon is AUG. AUG. It codes for methionine. Methionine is an amino acid. So how can I know that the AUG codes for the methionine? Because this particular tRNA has the anticodon UAC, and carries this methionine. So always the first protein in any protein sequence, protein chain, is the methionine. Why? Because the start codon is AUG. Then the large ribosomal subunit will join the small subunit, and it will start. I'll show you now the two sides of here we have the P side, and we have first, this is the ribosome, and the first tRNA will just go to the P site. Okay, so A site will be available for the elongation. So the elongation is the second step of translation, and it's actually the step after the initiation. Elongation, that I elongate this polypeptide chain, okay, adding more amino acids to that polypeptide chain. The polypeptide chain is the chain that has all the amino acids that I am adding together, just adding them, bonding them to the previous with a covalent bond or a peptide bond. Here, that this is the ribosome, okay, so the ribosome has a P site and here A site. It will start the tRNA. The tRNA will come here to the A site. The amino acid will be connected to the previous amino acids by a covalent bond. The polypeptide chain connected to the polypeptide chain by a peptide bond. Then the tRNA will move to the P site, leaving again the A site to the new one. Then a new one will come here, A site, then and so on. Okay, so this is the elongation process. Here a slowly process. This is the P site and this is the A site. So it will come, the new one will come to the A site, and when the amino acid is connected, it will leave and it will go to the P site, so the A site will be available for a new tRNA for new elongation. The last step is termination. So the last step here, I came to the stop codon and the polypeptide chain is released. The two subunits of the ribosome are separated and released, and everything is done. It's already completed the translation process. Now the ribosome is ready for a new mRNA to be translated. So guys, to sum it up, in the nucleus we have the transcription, the first. So this is the nucleus, and we have here the first step, the transcription. Then outside we have here the translation. The first step, the transcription, here we have from the DNA, we will just get a part of the DNA and we will just make a copy from this one strand of the two DNA strands to give that mRNA. Then the mRNA will go out after splicing. It will go out for the translation. It will be coded in the ribosome into proteins, amino acids, then it will give the protein. Thank you for watching, and just wait for the next video. Subscribe and like. Thank you so much.